Hello students, welcome to EPG Pathshala. I am once again Dr. M.N. Gupta, Emeritus Professor at IIT Delhi. Today we are going to talk on a module which is titled Types of Antigens and Immunization Procedures, and this again belongs to the paper called Immunology. In immunology, we talk of antigens and immunization in two different contexts. The first context is in vivo, the second context is in experimental immunology. It is always important to remember these contexts, otherwise it can lead to some confusion. We immunize animals to raise antibodies. We also immunize or vaccinate people. Immunization and vaccination are used as synonyms. When an animal gets exposed to an infection, the infectious agent is the antigen. This is purely a question of usage of terms. The objectives of this module are to learn about various types of antigens which we use in experimental immunology. Similarly, variety of antigens same way are also encountered by the immune system of the animals. We will also learn that what are the various types of immunization procedures, and we will also try to understand the features of three types of acquired immunity. So the concepts which will be basically covered in this module are that we will try to look at the various antigens of the microbial origin. In the previous module we had learnt about innate immunity. Here we are going to focus more on the acquired immunity. Acquired immunity is of three types: active, passive and adaptive. Microbial organisms have diverse types of antigens. The acquired immunity again has three roots, and the immunization procedures have to be viewed in the context of why immunization is being carried out. The preferred method of immunization against different infectious agents depend upon the nature of the infectious agents or the disease. Let us look at the proteins which are frequently used for immunization. In a later module, we will discuss what makes a particular substance a good antigen or a bad antigen. Proteins in general are frequently used for immunization because they are good antigens. In much of experimental immunology, we mostly deal with proteins and their conjugates as experimental antigens for this reason. Important microbial antigens are proteins or protein conjugates like clostridial toxins, bacterial flagella, viral capsids, protozoan cell membranes, snake venoms, serum proteins, milk proteins and hormones are also used as antigens for immunization. Antibodies themselves, being proteins, constitute important antigens in experimental immunology. Let us look at the bacterial antigens. While bacterial cytoplasm contains many antigenic substances, it is the surface antigen which are important in provoking an immune response in vivo. The cell wall is the first important surface antigen. It consists of peptidoglycans in case of gram-positive bacteria and is made up of polysaccharide lipoprotein structures in case of gram-negative bacteria. Composition of the trisaccharide is important in determining antigenic property of the organism. We can exemplify it by looking at the Salmonella. Salmonella has 2000 species distinguished on this basis. The three types of antigens, that is the trisaccharide units with glucose as the terminal unit, paratose as immunodeterminant sugar, and mannose as immunodeterminant sugar, these all act as antigens. Let us closely look at the cell wall of gram-negative bacteria. Cell wall lipopolysaccharide of gram-negative bacteria are endotoxins. In these cases of gram-negative bacteria, the oligosaccharide part is attached to lipid A, and the repeating trisaccharide units are the important antigenic structure. The lipopolysaccharide O, for example, in itself is an antigen. So is the lipid A, which is an endotoxin, which is also important in the entire scenario of the infection. The detailed structure of the cell wall of the gram-negative bacteria is shown in this slide. The cell wall has an outer membrane, and then it is a peptidoglycan structure, followed by plasma membrane, and finally by the periplast. The lipopolysaccharides form outer part of the outer cover after the cell wall. Some bacteria are enclosed by capsules, which are either protein or polysaccharide in nature. Capsular antigens are called K antigens. Pili or fimbriae are called F antigens. Bacteria flagella consist of protein called flagellin, and these antigens are called H antigens. Proteins secreted by bacteria or obtained by autolysis of bacteria are called endotoxins. The antibody formed against all these bacterial antigens have protective function. Viruses also act as good antigens and provoke immune response. That is because they have a protein coat which is called capsid. These capsids are good antigens. Early unsuccessful blood transfusions between unrelated individuals led to the realization about the A and B antigens. These various blood groups arise from the fact that different erythrocytes have different surface antigens. These constitute important examples of the cell surface antigens of the mammalian cells. Because these erythrocytes have different surface antigens, their response to the various antibodies are very selective, hence the need for blood matching in case of the blood transfusion. Let us introduce another very important class of antigens, which are called histocompatibility antigens. Nucleated cells have many cell surface antigens. Most important ones are these histocompatibility antigens, which provoke strong immune response and are called major histocompatibility antigens. These play a very important role in immune responses, and we will have occasion to repeatedly refer to them in more detail, in fact, in the future lectures. Autoantigens and superantigens are two different classes of antigens. Autoantigens are the antibodies produced in response to the normal self antigens, whereas superantigens are antigens which provoke an exceptionally high T cell response. Some antigens are commonly used in experimental immunology as these are relatively cheap and are available in pure form and large quantities. Let us have a look at the commonly used antigens. This list consists of some commonly used antigens in experimental immunology. Endotoxins from E. coli, bovine serum albumin, flagellin, hemocyanin, which is the blood pigment of keyhole limpet, IgG, that is immunoglobulin G, lysozyme, sheep red blood cells, and also TGAL, which is a synthetic antigen consisting of tyrosine and glutamate linked to the alanine side chains on a lysine backbone. These are all commonly used antigens. Immunization is of three different types: active, passive, and adaptive. In active immunization, the animal is administered an antigen. In passive immunization, preformed specific from one animal is transferred to another non-immunized animal. In adaptive immunization, the immunity is also transferred from one animal to another, but by the transfer of the immune cells themselves. Let us look at some of the features of these three types of immunizations. All the three types have different functions. In active immunization, protection is not immediate, but it is often long lasting, and re-immunization can be carried out. What we call vaccination is often an active immunization process. In passive immunization, immediate protection is possible because preformed antibodies have been injected. However, it is short lasting as these preformed antibodies which have been transferred are degraded after some time. James Gowans originally used adaptive immunization to determine the role of lymphocytes. Here lymphocytes are transferred. Such transfer is possible, of course, only between genetically identical donors and recipients, otherwise this itself will give rise to strong immune response. We mentioned vaccination. A successful vaccination leads to a long-lived state of protection against the specific immunizing pathogen. If this immune protection can be transferred to a normal synergenic recipient with serum from an immune donor, then immunity is mediated by antibodies. Such immunity is called humoral immunity, and the process is called passive immunization. If immunity can only be transferred by infusing lymphoid cells from the immune donor into a normal syngeneric recipient, then the immunity is called cell-mediated immunity, and the transfer process is called adaptive transfer or adaptive immunization. As we already mentioned, passive immunity is short-lived as the antibody is eventually catabolized, but adaptively transferred immunity is mediated by immune cells which can survive and provide longer lasting immunity. Passive immunization is useful for immunocompromised individuals who have deficient immune system responses. Usually, Ig is given either intravenously or intramuscularly. A well-known example is the transfer of antisera from horse against tetanus to humans. It is usual to administer equine fragments to minimize its antigenicity protection of fetus by maternal antibody. and intestinal absorption of cholesterol antibody are examples of natural passive immunization. This figure gives us the detailed classification of the acquired immunity and provides a clearer picture of the various ways the acquired immunity may be obtained. Protection of the fetus by maternal antibodies or by administering artificial immunoglobulins comprise the passive acquired immunity. Active immunity can be obtained through natural infection or by administering artificial agents like living or dead organisms, metabolic products like toxoids, or genetically engineered antigens and other synthetic antigens. Sometimes animals are given active immunization to raise antisera. Rabbits, goats, sheep and fowls are generally used. The unrelated species has to be used. A far removed animal or bird from the source of the antigen on the evolutionary tree is the best immune response. Differs from animal to animal. So initially several animals should be tried. So if the volume required is large, goat or sheep are preferred as these are larger animals. Expenses like cost of animals and their housing sometime have also to be necessarily considered. In experimental immunology, the immunization injections may be given simply intradermally, that is in the skin, or they may be given intramuscularly or subcutaneously. Single site or multi-site injections may be used. Multi-site injections provoke the immune system by different routes and generally result in better titers of the antibodies in the blood of the animal. Under the best conditions, that is the optimized conditions, antisera can be raised with antigens in microgram amounts. Mixing of substances which are called adjuvants with antigens help in obtaining better immunization. Proteins which show less evolutionary divergence between mammalians turn out to be poor antigens. That is because their foreignness is very limited in terms of their structure. Examples of such proteins are cytochrome C, actin and myosin. There are two approaches by which you can overcome this hurdle in the case of such proteins. One can raise antibody in fowls. You can also cross-link to form homopolymers or heteropolymers by conjugating these proteins with ovalbumin. This normally produces a better titer of the antibodies in the case of these proteins which show the less evolutionary divergence. As an illustrative example, we can look at the excellent titers which have been obtained for the antibody against the rabbit casein polypeptides. In such cases, the proteins can be linked to the micron-sized particles of agarose. The trade name Cephalose 2b is for some of the agarose particles. So the conjugates like Cephalose 2b, albumin and casein have been used to improve the antigenicity of caseins. First, the Cephalose 2b is activated by cyanogen bromide. The albumin is linked to it. Albumin now has many free amino groups. The cross-linking between the rabbit casein polypeptide and albumin can be created by linking free amino groups present on the two proteins via glutaraldehyde, which acts as a cross-linker. Finally, you get these C-Pharos 2b albumin casein as a conjugated antigens. Immunization schedules should be adhered to. The non-immune serum before immunization should be collected for running controls. Immunization should be repeated at regular intervals. Preparation of low affinity adsorbents does not require high titer antiserum. Immunological purity is different from biochemical purity. This should be kept in mind while assessing antibody preparation. In general, shorter immunization schedules contain fewer antibody to contaminating antigens. Trial bleeding should use simple methods like rabbit ear veins. In case of sheep or goat, venipuncture of the jugular vein works well. Preparative bleeding may require local anesthesia. At least 2 to 3 weeks should intervene between successive bleedings. The humoral response comes in after a lag period. Initially, IgM is produced, but class switching takes place and IgG response is higher, but is slightly later in the time frame. The secondary response after giving a booster dose, which is a repeat of the primary response events, consisting of switching over to IgG, in this case is quicker and the response is more intense. Let us look at the priming immunization and primary response. The first immunization is sometimes referred to as primary immunization. The humoral response of the immune system in terms of antibody protection is called primary response. The primary response itself has 4 phases. During these different phases, both the concentration and the nature of immunoglobulin changes. So, let us look at the 4 phases of the primary response which can be identified. First, of course, is the latent or lag phase. It is the initial 2 to 3 weeks period after the first immunization. During this period, antibody is not detectable in the serum. Obviously, if less sensitive methods for antibody detection is used, the lag phase may appear to be longer. Second is the exponential production phase, when the antibody concentration in the serum rises rapidly. Third is the steady state, when the antibody concentration reaches a steady state. This is the state when the rate of production of the antibody and its degradation are balanced. Finally, there is a decline phase when immune response slows down and the antibody concentration declines rapidly. Generally, only one kind of immunoglobulin called IgM is synthesized during the primary response. If another kind of immunoglobulin IgG is synthesized, its formation is detected only after IgM concentration has nearly declined. Just like the primary response, the secondary response can also be looked upon in terms of its distinct phases. In the case of the secondary response, the lag phase is shorter, almost cut down by half. That means the secondary response is quicker than the primary response. In this secondary response, IgM may or may not be detected. If it is detected, it is soon replaced by IgG kind of antibody. Serum contains much higher amount of the antibody. The secondary response persists longer and sometimes can last as long as months or even years. This is the kind of response where other kinds of immunoglobulins or antibodies may be formed. The capacity to give this secondary response may persist for years in humans. This is also called memory or anamnestic response. It is this response which serves as the basis for human immunization programs. So, students, let us now summarize what we have learned in the present module. First of all, we have learned that the immunization may be intentional, as in experimental immunology, or when we are trying to vaccinate somebody, or it could be accidental, like when infection happens. Antibody in response to these immunizations, whether they are intentional or accidental, depends basically upon the type of antigen. Again, in experimental immunology, or if necessary for medical purposes, immunization may be active, passive, or adaptive. We have also learned that in active immunization with experimental animals, immunization schedule, bleeding schedules, and few other factors are important. Active immunization consists of primary response and secondary response. We have discussed immunization in simple terms, basing our discussion purely in terms of humoral response. This in itself is not the whole story, because as we have been indicating, there is also a cell mediated response. However, humoral response in itself is the result of complex interplay between various other components of the immune system. Thank you.